Welcome to Mex Academy. In this video, we're going to do Introduction to Life Sciences. The content which we're going to cover in this video, we're going to look at the structure of the life sciences as in how the then we'll look at how one can life sciences test or examination, and then looking at the structure of the life sciences paper. Life science paper is divided into paper 1 and paper 2. And here are the topics which are covered under paper 2, which is code of life, meiosis, genetics and inheritance, and evolution. Then in paper 1 there's meiosis, reproduction in vertebrate, and human reproduction in responding to the environment, human endocrine system, homeostasis, and responding to the environment in plant, and then human impact. Okay, this topic is done in grade 11. Continuing on the structure of the paper, okay, by paper 1 and paper 2, we mean that life science on its own, okay, life sciences exam, that it has paper 1 and paper 2. And these papers, they're not written at the same time. So you can write paper 1 examination on the 12th November and write paper 2 on the 15th November. They're written in different time. Okay, but on each paper there are 3 sections. First section, and section A requires short answers. These are multiple choice questions giving the biological terms of the statement given. Then section B has variety of questions. In total, there are 80 marks, and in section C, it's the essay question, which is 20 marks. So in paper 1, you're going to get these sections, and then in paper 2, you're also going to get these sections, but on the topic which relates to that. Now, preparing for life sciences test or examination, it is important to go through the notes and understand them, and then do examples on that particular topic, and do the tutorials, and then do the past papers. That by doing examples, tutorial, and past papers, you are familiarizing yourself with what are going to be asked. And in doing also the say how essays are asked, which is the question that is asked in section C. Then it is important to love this subject, not only the subject, all of the subjects. It's important to love them, because if you love them, you're going to enjoy doing it. Really, really important to love the subject. Which now starting with our okay, we do have videos of nucleic acid, so you can go through them. Also in this video we're just gonna summarize the nucleic acid, but you can also go through the videos which you posted previously about nucleic acid, and then we'll then go into do examples in this nucleic acid. Okay, but we'll do examples in this separate video. There are 2 nucleic acids found in a cell. We have DNA and RNA, and then these are made up of the building blocks or monomers called nucleotides. Please note this pollute, and also nucleotide consists of phosphate group and nitrogenous base. Note which sugar makes DNA and which sugar makes RNA. Which phosphate group? Okay, for the phosphate group, it is the same for DNA and also for RNA. For nitrogenous base, they are different. So it is important to look at them to be able to spot the difference in the nitrogenous base for RNA and that of DNA. Let's go to the next slide. in DNA, there is the phosphate group and also in RNA. In terms of sugar, the sugar which is found in DNA is deoxyribose sugar, and in RNA we have ribose sugar. And the nitrogenous bases which are found in DNA are adenine, thymine, guanine, and cytosine. And in the nitrogenous base which are found in RNA are adenine, guanine, cytosine, and uracil. So the different nitrogenous base is uracil in RNA and thymine in DNA. Then DNA is double-stranded while RNA is single-stranded. We're going to show this on our next slide. But it's also important to know that adenine bonds or goes with thymine, and cytosine goes with guanine, and in RNA it's adenine and uracil and cytosine with guanine. Let's go to our next slide. Here is our next slide. It is showing that DNA is double-stranded. Here is the first strand, and here is the second strand. For RNA, it's only one strand, single strand. Here are the nitrogenous base, which is adenine, and here is thymine. They are combined by a weak hydrogen bond. It is important to also note that it's weak hydrogen bonds which combine or which connect the nitrogenous bases in DNA. And here is the phosphate group and the deoxyribose sugar for the sugar, and then these are the nitrogenous bases. As we said, nucleic acids are made up of the nucleotide which consists of sugar, which consists of phosphate, sugar, and the nitrogenous base. It is important to note that and to be able to differentiate between the phosphate group, because you might be given a question which will point here, the other one point here, and the other one point here, and then you ask them, what is this? You have to be able to see that it is the phosphate group, and this one, it is the sugar, which it will differ. It will differ if you're dealing with DNA, it's going to be deoxyribose sugar. If you're dealing with RNA, it's going to be ribose sugar. So, okay, it is important to be able to spot or to see the difference between DNA and RNA molecule, because in an exam or test you may be given a structure or a molecule which may be DNA or RNA. If you're not able to see whether it's RNA or DNA, you're going to answer the questions wrong. Know the difference in these two molecules. This picture introduces us to DNA replication. We're going to okay, DNA replication is a process where DNA molecule makes a copy of itself, and this process occurs before mitosis or meiosis begins. So the following are the steps of DNA replication. So step number one is that the double helix unwinds. So DNA in its state, it is found in a double helix. So for DNA replication to occur, the DNA needs to unwind. Let's go back to our picture. Here is our picture. So this is the state where the DNA is found. But after this state, the DNA is starting to unwind. Here is DNA. This what is unwinding. Here we can say confuse my handwriting, but DNA here unwinds. Okay, let's go to our next line. Weak hydrogen bonds between nitrogenous bases break and two DNA strands unzip or they break. Okay, we said that between the nitrogenous bases there are weak hydrogen bonds. So we're saying that the weak hydrogen bonds they're going to break, and when they break, we're going to get two strands. So DNA, as we said, DNA is it is double-stranded. So as it is double-stranded, when the weak hydrogen bond break, these two strands they break, then we get two strands. Example, gonna be a strand like this. This is the strand, and here. Okay, let's take from here. We have G, which is guanine. So what is going to separate the weak hydrogen bonds between the nitrogenous bases are going to separate. No, the weak hydrogen bonds, they're going to break. As they break, they are separating the nitrogenous bases. We have guanine, let's take it from here. Then we have thymine, and then we have guanine again. And then on this side, we have cytosine connected to guanine. Then we have adenine, which was connected to thymine, and then we have cytosine, cytosine connected to this guanine. We have our two strands as created as the result of break. Moving on to the each original DNA strand serves as a template on which its complement is built. So now on these two strands, complement are going to be built like this. Guanine connects with cytosine, thymine with adenine, and guanine again. So weak hydrogen bonds. And then in the second one, also the complement is going to it's E for thymine, and then guanine. And then here we can say this is DNA molecule number 1, and this is DNA molecule number 2. We have to remember that we had one DNA molecule created as a result of the weak hydrogen bonds doing what? And then 3 nucleotides build a strand on each of the original 2 DNA strands by attaching to their complementary nitrogenous A to C and C to G. This results in 2 identical DNA molecules. Each molecule consists of 1 original strand and 1 new strand. We have this here. So first thing what happens first is that we need to understand that DNA in its state, it is in double helix. And then when DNA replication is to happen, the DNA in double helix has to unwind. When it unwinds, the weak hydrogen bonds, they're going to break. When they break, we get DNA strand, get two strand. And then from this strand, floating the nitrogenous bases are going to attach to this strand. As they attach, they are building another DNA molecule. And then as a result, we're going to get DNA molecule process. That's DNA replication process where DNA makes copy. This question may be asked in the essay question, which is section C. Please know its way. And then looking at the significance of DNA replication, it's also may be asked just to remember. It doubles the genetic material, so it can be shared between the resulting cells during cell division. Cell division, which may be mitosis or meiosis. As we said, that it occurs for meiosis or mitosis genes. Point number 2, it results in the formation of cells during mitosis. DNA profiling. Every person except identical twins has his or her unique DNA profile. So DNA profile is the arrangement of black bars which represent DNA fragment of the person. So like a profile on Facebook, like a profile on Instagram, we as humans we also have DNA profile. And our DNA profile is from human, it's not identical. For twins, for identical twins, then identical to them. Here is how the DNA file looks like. But they are black. Yes, DNA profile, something like. So why do we need this? They are used to identify criminals, identify dead bodies, identify relatives, and identify anything. Okay, let's take an example on how they are used. To identify criminals, let's say there was a crime scene, or let's say there was a break-in. There was a break-in. There was a break. In the break-in, blood is found, blood which is in the break, and this blood is taken by the police to do or to find the DNA profile. Okay, a DNA profile will be found using the blood as the crime scene of break-in, and then the police they go arrest suspect, and there's gonna be, let's say there's suspect number 1 and number, and then the police they're going to do the research on the DNA profile of this crime of this suspect number 1. Okay, this is the DNA profile of suspect number 1, and for suspect number 2, their DNA profile may be something like this. There's already 3 bars in suspect number 2, so meaning there's a big possibility that suspect number 2. Okay, let me also. Okay, here is the blood for suspect number 1. What happens here that we had the what? breaking. The breaking, there was the blood. This blood, the DNA profile was found. So the police have this has this evidence, or they have this on their side. They have this DNA profile. They arrest two people, the suspect number one and suspect number two. So now they have to match the DNA, the DNA profile which they found at the scene with the DNA of suspect. Looking at the DNA profile of suspect number one, is it the same to the DNA profile which was found? Yes, they are the same. So we can say that suspect number one was at the crime scene, so maybe the one which committed the crime. He will be arrested, and number two, and that's how it used dead bodies or to and also relatives. Anything you're gonna have parent, not gonna be in this case, but it's gonna be a parent. It's gonna be parent number one, father one, and parent number two. And this one, it's now going. And this one, we can say then it's now going to be DNA of a child. It's the DNA profile of a child. And then in this case, it's for parent number one and parent number two. Then you can check if the DNA of the parent one, is it the same? Of father one, is it the same to that of the child? Father two, is it the same to that of the child? From this, we can see that parent number one and the child DNA profile are the same. Then we know that number one or father number one is the father of. Then protein synthesis also may be asked in the essay. Protein synthesis is a process where, okay, it can be asked in the essay. This is the process by which proteins are made in each cell of an organism from enzymes, hormones, and new structures for us. So there are three forms of RNA. So looking at these forms of RNA, we're going messenger RNA and also ribosomal. Messenger RNA carries the message. RNA transport the amino acid. Okay, here is summary of the picture of protein synthesis. You know picture, you're ready for the act. Okay, protein synthesis is divided into two main processes: transcription and translation. Let's look at transcription. Place in the nucleus. Okay, now that's the process that by the DNA unwinding, and then it split. One DNA strand acts as a template for forming the messenger RNA. Three nucleotides are arranged to form messenger RNA according the DNA template. This process is called transcription. Then messenger RNA leaves the nucleus through a pore. Okay, let's go to our picture. Okay, we said that, okay, here is DNA. It is in a double helix form. Okay, number one, we said it's going to what? Going to unwind. Okay, here is the DNA. It is unwinding. And then said that one, one DNA strand acts as a template of forming messenger RNA. So here our strand. This is strand one as a result of the DNA unwinding and splitting. And then here is strand number two. And then we said one strand will act as a template of forming messenger RNA. So here is this strand which is forming messenger RNA that is being formed. So how is messenger RNA formed? Let's summarize it here. If Here we said our DNA is going to split if our DNA, if one of our strand, which acts as the template for forming messenger RNA, has this nitrogenous base, there's adenine and thymine and cytosine. And the messenger RNA will be formed also adenine, the complement will be uracil, thymine, the complement will be adenine, cytosine, guanine will be the. So this one is now messenger RNA. So RNA, this one was the DNA strand which is a template forming messenger RNA. From this, it is important to note that since it's messenger RNA that we are forming, it's going to have uracil instead of thymine. If it was we're forming another DNA, or if it was a replication, it was going to be thymine here. But since we're forming messenger RNA, and from this we get our messenger RNA. Here is our messenger RNA, and this three, and this three, and this three, they are called codons. So we divide the nitrogenous base which is found in messenger RNA into triplets, and then we call each triplet codon. So this is a codon, this is a codon, and this is a codon. Then after messenger RNA is formed in the nucleus, it's then going to leave the nucleus, and then we're going to start our second stage, our second process of protein synthesis is translation. The stage B, and it takes place when messenger RNA in the cytoplasm is to the ribosome. So it takes place in the cytoplasm of the ribosome. So here is messenger RNA. It attaches to the ribosome, and each transfer RNA brings a specific amino acid. Then messenger RNA, this is called translation. The amino acids are linked together to form a pattern. Okay, we have messenger RNA here with the codon, as we know that this is codon. And then transfer RNA. RNA also have these three triplets. These three are called anticodon. So they're the complement of the codon. And they carry the amino acid. Each transfer RNA carries amino acid. Amino acid. Amino acid. So, okay, let's say we have a codon. Let's have a codon here. Okay, codon. Our codon has A, U, and C. Then, okay, here we have A, U, and C. Then our transfer RNA, in order to bond with this, need to have O, A, and G. And then this other transfer RNA also come here. Let's say we have C and C. This one need to come with G, G, and then this one will attach here, and as it attach here, it will this will bond, and as they bond, the amino acids from here will join here, and as they join here, they are forming a peptide chain, and that is when the protein is being formed. So we say the amino acids are linked together to form a particular protein. So this is when the amino acids are being linked here, they're being linked here, and they're going to form a protein, as we know that amino acids are the building block of a protein. That's the end of chapter 1. Note, examples will be done in the following video. Please don't forget to subscribe, like, and remember to write your question on the comment section. And also remember to share the link of help with your friends. They may also watch this. Thank you for watching.